right, the last speaker of our session is given by Dr. Wei Yan-ying from University of Carolina at Chapel Hill. And he will speak to us on tracking synergistic and redundant neural interactions from preterm to term infant brains. Let's welcome.
Okay, hello everyone. My name is Wei Yan-ying. I'm from the University of North Carolina. And it is my great pleasure to have a chance to represent our research team and present our recent study about tracking the synergetic and redundant neural interactions from preterm to term infant brains. So I have no financial interest or relationship to disclose about this presentation. And okay, let's get started. Over the past decades, brain functional connectivity has been widely used for studying brain functions. It has led to many important findings in both healthy and diseased populations. And traditional seed-based graph theory or dynamic functional connectivity have been reported in lots of studies. However, traditional connectivity methods mainly look at the pairwise connection, and which means that they may miss out some hidden, more complex information within the both signal. So in this study, we use a newly developed information decomposition method to go beyond this traditional functional connectivity method. And this allows us to capture more both synergetic and redundant interactions between brain regions, and therefore offering us a more richer picture of how brain regions work together. So let me first explain what we mean by synergy and redundancy. A very simple example comes from the visual system. Each of our eyes have its own visual field, and there is a large overlapping area between them. This overlapping area is actually represent the redundant information of our visual system, since both eyes are seeing the same information. In contrast, the sense of the visual depth is actually the example of synergetic information of our vision. It is something that we can only obtain by combining the input from both of our eyes, and no single eye alone can capture this depth perception. In short, synergy is actually the actual information you can get when combining signals together. In neuroscience field, brain synergy can represent the process of functional integration. On the other hand, redundant information is the shared information from multiple sources, and it contributes to the functional robustness of the brain system. So based on the recently proposed integrated information decomposition theory, the information between two brain regions can be further decomposed into three components. That is the synergetic information, unique information, and the redundant information. Lupi and his colleagues has provided very novel insights into synergy and redundancy, and they have provided some explanations to the function roles of the synergetic and redundant interactions. Specifically, they have demonstrated that the synergetic interactions tend to support the integrated process and complex cognitions of human brain. And the redundant interactions, on the other hand, support modular sensory motor processes. And in our study, we want to extend their study and focus on how this brain synergy and redundant develop during very young infancy. So in this study, we use a publicly available DHCP dataset, which includes preterm infants aged from 26 to 45 gestation weeks. Some of the preterm infants also have the term equivalent scans. That's why they have some very older scans at 40 to 45 gestation weeks. And this dataset also includes a subset of full-term infants. And in total, we have included 623 subjects with 701 fMRI scans in this analysis. And for this analysis, we still apply the adult parcellation, since there is still no widely accepted functional atlas for infants, especially for preterm infants. And we combine the 200 ROI shuffle cortical atlas with a recently published subcortical atlas comprising of 32 regions. And these regions were further grouped into 8 functional networks, including typically named 7 networks proposed by Yao's group and 1 subcortical network. Here is the example. This is on the left of the slide is the example matrix of synergy from a subject scan at 34 weeks. As we can see, at a very young age, synergetic interactions across the whole brain are generally very low, except within the sensory motor network. When we look at the development trajectory of whole brain synergy, we saw a very clear pattern. The synergy starts from near zero, and then after around 35 weeks, they start to show a rapid increase. And they continue to increase until 42 weeks, and followed by a stabilized pattern. This development pattern is widely observed in all the functional networks except the subcortical network. While most of the networks showing increase and then stable pattern, the subcortical network display a decline around 42 weeks. Meanwhile, along all the networks, the sensory motor and the dorsal attention network tend to have the highest peak synergy values compared to other networks, while the limbic and the subcortical network have a pretty low synergy value throughout the development process. For the whole brain redundancy, similar pattern was also observed. The redundant interactions are pretty low in the 34 weeks. However, within sensory motor area, the redundant information is starting pretty high at this age. For the development trajectory, we observe a developmental transition starting at 33 weeks, which is relatively earlier than the synergy transition. And the redundancy gradually increased up to 40 weeks and followed by a decline after 43 weeks. This pattern was also observed in the sensory motor network, salience network, and subcortical network, while all the other networks follow the increased and stable pattern. Notably, the sensory motor network still maintains the highest redundancy, peak redundancy among all the networks. And on the other hand, for the basic function networks such as the visual network and the sensory motor network, their first transition age is pretty early compared to the high-order functional networks. And more importantly, we also observe that the transition age in redundancy are relatively early than those observed in the synergy patterns. So for the subset of preterm infants that has the term equivalent scans, we further compared their synergy and redundancy with those of full-term infants. Significant differences were observed among the 5 age groups. Specifically, the post-hoc analysis confirmed that full-term infants have significantly higher synergy and redundancy compared to all the other preterm infants. And meanwhile, infants born between 35 to 40 weeks also have significantly higher redundancy compared to infants born before 35 weeks. So in addition to this result, we also want to highlight some potential factors that may affect the estimation of synergy and redundancy. Since the information decomposition framework is based on Shannon entropy, factors that may influence the signal-to-noise ratio of the time series data may affect the estimation of brain synergy and redundancy. These factors may include, but are not limited to, the different imaging techniques, pre-processing steps, the length of time series data. And here is a quick example about the influence of time series data. We found that synergy value will increase along with the increase of number of tier, while the redundancy value will decrease along with the increase of tier. And both of the metrics began to stabilize at around 800 tier. This 800 tier is much longer than clinically applied fMRI scan or other public available data sets such as Baby Connectome Project, HCP aging, HCP development. Therefore, to help better understand and interpret brain synergy and redundancy in different data sets, we need to systematically evaluate these factors so that we can know better about whether it's due to the bias or truly meaningful. So to conclude, this study revealed the developmental pattern of brain synergy and redundancy at both whole brain level and the network level. Most of the trajectories follow the increase and stable pattern, while only a small subset of them showing a pattern that includes subsequent decline. Importantly, we found that the transition age from 0 to rapid increase in redundancy occurred early than synergy. This suggests the potential importance of establishing robust network interactions before the emergence of the synergetic interactions of human brain. On the other hand, sensory motor network also had the highest level of both synergy and redundancy. This result highlighting the potential developmental priority of sensory motor functions during very early infancy. This study also highlights the potential functional difference between full-term infants and preterm infants at their term equivalent age. And this work was actually led by Professor Willie Lin from UNC Chapel Hill, and with a lot of contributions from many collaborators, including PhD students, visiting scholars, and faculty members of UNC. And although I'm not the first author of this abstract, but I will do my best to answer any questions you may have. Also, our poster number is 1007, and we would be happy to have you stop by to discuss these findings. Thank you very much.
Thank you, Wei Yan, for the very nice talk. We have many questions for the audience, but I'll just pick one of the interesting one. Is it possible that the increase in synergy and redundancy is due to that you're having more data at a later age compared to the earlier age?
Actually, based on the distribution of the data, we still think that it's truly due to their developmental process instead of we have more data. Because we also test the result in later BCP age, we also found some increased patterns in both synergy and redundancy. So it means that the developmental is relatively continuous development from very early infancy to maybe later ages.
So did you control for the sample size at each
we use all the data.
Okay. And I'm also curious that since you said at the beginning, the synergy, redundancy, and unique information, they add to one. If the synergy and redundancy increase with age, does that mean that unique information decrease with age? Is it possible?
It depends, because the mutual information between two brain regions also may increase with age. So it may need to test the unique information, whether they will have different age development patterns. But based on another study performed by our collaborators, the proportion of synergy and redundancy keeps increasing during this age range. Therefore, maybe the unique information is pretty different. Yeah.
All right. We're sharp on the time, so thank you once again.
Thank you, everyone. Just a reminder, there's a review of the session on the app. If you could do that, that would be awesome. Thank you.